Hello, everyone. My name is Joanna, and I am the Director of Knowledge Mobilization at Canada's Stem Cell Network. It's my pleasure to welcome you all to the third session of the four-part virtual training series, Write It Right, Advancing Your Approaches to Scientific Communication, that we are pleased to co-host with Project MaVen. This training series is designed to help equip highly qualified personnel with advanced approaches and practical skills for scientific writing within their academic career and beyond. Each of the four sessions focuses on a different aspect of scientific writing. Today's webinar is focused on communicating science clearly, how to write for non-expert audiences. SCN is a not-for-profit organization supporting stem cell and regenerative medicine research through training, knowledge mobilization, and funding opportunities that accelerate the translation and commercialization of stem cell and regenerative medicine innovations. We're excited that you've joined us for today's webinar, where our panelists will discuss how to identify key considerations for writing about your science for non-expert audiences, simplify complex ideas without losing accuracy, and tailor your message to engage broader audiences. This is one of many training opportunities that SCN hosts throughout the year as part of our training program. I encourage you to check out all of these opportunities on our website at stemcellnetwork.ca. With that, I'd like to hand it over to Megan Wright, our representative from Project MaVen, to say a few words.
Thanks so much, Joanna, and welcome everyone. My name is Megan. I'm the project manager for Project MaVen. Project MaVen is a multi-institutional initiative. It's led by Dr. Michael Laflamme, who's a world-leading researcher in stem cell-based therapies for heart disease, based out of UHN's McEwen Stem Cell Institute. Project MaVen, though, is funded by a federal tri-agency funding program called the NFRFT, which stands for the New Frontiers in Research Fund Transformation Stream. It's a really interesting program. You can learn more about either at our LinkedIn page or the website, both of which are linked there with QR codes. So before we get started, I'd like to go over a few housekeeping notes. In terms of the format for today's session, each of our panelists will give a brief talk on their strategies for communicating science clearly to non-expert audiences, and then this will be followed by about 25 minutes of panel discussion moderated by Joanna. Then we'll open the session to you, our attendees, to ask your questions to the panelists. You can put any questions you have throughout the session in the Zoom Q&A box, which you'll find at the bottom of your screen. And then if you see a question you like or a question that you were hoping to ask that somebody's already asked, you can upvote that question. And that'll bring the question to the top to make sure it gets posed to the panelists and answered. The session is being recorded up until that audience Q&A section, which point the recording will be stopped. Okay, and with that, I'll hand it back over to Joanna and we can get started.
Great, thank you, Megan. Before we begin our panel discussion, each panelist will address a different learning objective for today's webinar. Up first is David Kent, who will speak to key considerations when communicating your science to non-expert audiences. Dr. Kent obtained his PhD in genetics at the University of British Columbia and is currently a professor at the University of York in the UK. His lab has taken novel approaches in single-cell biology to better understand blood stem cells in humans. David has a keen interest in improving the way that we communicate science and educate and train scientists, including launching a column for early career researchers on universityaffairs.ca and writing for the CCRM Signals blog on regenerative medicine. He is currently a co-investigator on a Wellcome-funded Valuing Voices project to improve research culture. The floor is yours, David.
All right, great, and thanks so much for the invitation to join you all today. It's been a long time since I've done something with the Canadian Stem Cell Network. As you can tell by accent, I'm not from the UK originally. I'm born and bred in the Canadian stem cell environment. So yeah, it's nice to be back. So I'll kick off with by sharing my screen and with a short talk about what I try and do, at least when I'm communicating science to non-experts. So as introduced already, I'm currently at the Center for Blood Research at the University of York, and along the way, I traveled from Newfoundland originally, and so started here and did my BSc in Genetics and English Literature at Western, and then moved out to do a PhD in Vancouver with Connie Eves at the Terry Fox Lab, which is where I was introduced to the stem cell network and attended my first stem cell network meeting. This is bad in 2003, so quite some time ago. And then I decided to move 4.5 hours flying closer to my parents by moving to the UK. Right, we live in a ridiculously big country in Canada, and it's a long journey, but actually it's quite a short flight across the ocean from the UK. And I did a postdoc with Tony Green at the University Cambridge, where I really switched from sort of mouse stem cell biology into understanding how things go wrong in humans. And then started my lab in Cambridge, and then moved to York 5 years ago now, and as introduced again, a really focus on single blood stem cells and how they go wrong in people. And more recently, we've been focusing a little bit on the physical biology of stem cells as well with some bioengineering approaches. So I think we're all in a sort of funny place when we try to go outside of our science, and I mean I started going outside of science from a very very early age with the English degree, but this sort of conflict with traditional scientists and how we communicate and what we do outside of the lab and how we balance that with doing things inside the lab, and you don't always get the nicest reaction when you do this sort of a thing, and certainly you don't know about the reaction you don't get. And often there are these sort of old school traditional views about what we should be doing and in terms of our public responsibility to communicate in science, and what it really means to be an academic, especially in the age of sort of the sort of what one likes to call post-expert phase of life. And that's a really scary thought for people who invest an incredible amount of time and dedicate their lives to generating evidence-based or evidence bases that we base decision making off of. I recognize for those on the call that are trying to push these sorts of careers in public engagement, outreach, science communication, it's sometimes frustrating to sit inside of the academy and have views posed against that, but it is a really critical thing that we do. Now, when I started writing, it was in the time when blogs were actually a thing, and there was quite a lot of them going on, and there were lots of really rubbish ones out there too. And I think really the sort of big problem is trying to find that sort of glorious space where the content is really valuable, and we like to push at that in some of the spaces that I've been writing in, and really try and pose these solution-based kind of articles about the science. And really a great example that inspired me when I was training in my PhD was from Rosie Redfield at UBC. She actually just got really annoyed about a paper and just started blogging about it because she, wasn't part of the review process, but read it and said, this can't be true. And then she went off and did some experiments about it, blogged about it, and then got a science paper 2 years after raising it in the public sphere. But just this idea that you can police the literature as a scientist was quite fun. That idea that we could really push back at things that we thought were maybe untrue. And so why do I write for the public? Well, it's that sort of kind of science-based stuff, but also I find that's a really fantastic way of keeping informed. And a lot of the stuff that we write about on university affairs, at least historically, has really been around funding policies and sort of the way that we think about how we educate and train scientists. And you don't often find time to read for that unless you have the excuse of needing to write about it. And so that's a really great way of staying on top of things. And really, I find it helps me push myself out of that sort of really, that comfort space of reading and writing about the things that you always or already know about. And then I think a lot of it comes from that sort of desire to make the world a better place than where you started from. And so sometimes there's this sort of frustration around how we train scientists, and then also how those scientists feel treated when they go through their training programs. And I know lots of you will be sitting in PhDs right now wondering sort of about the state of training and how we can make it better. So hopefully this will spur you on to write some things about it and try to get some activity in the right direction. So who or what did I write for? One of the big ones was Signals, and that was the blog started out of the Canadian Stem Cell Network and taken over by the CCRM eventually. And really, this is giving opinions as a sort of domain expertise of stem cell biology and regenerative medicine, gene therapy, that sort of space, and really sharing about new discoveries in that area. And probably the one that was most random was the black hole. And this is something that we started as a bunch of PhDs and postdocs when I was training in UBC. And we used to meet about the state of training and how you could get an undergraduate degree without any laboratory experience and how frustrating that was and how science or how funding policies were all backwards. And we just had opinions. And so we decided that we would try to mobilize some of that. And we started the Black Hole. And then that was eventually purchased by University Affairs. And we continued writing for it for nearly 15 years. So it's been a long time with University Affairs and a really great amplification of a lot of these ideas. And then more recently, I've done some work with a hematology specific journal, really around sort of the space that I operate in. And really, why I do this is both to filter the literature and focus on topics that are interesting and I can give that domain expertise on, but also in particular with the black hole is really trying to have that information get into the hands of policymakers. And this is where University Affairs is really, really fantastic because many of the university administrators read University Affairs. And if you're making comments on how parental leave policies should be or how research funding should operate and how institutions can get involved with shaping research culture, it's been a really fantastic place because it really amplifies the message. And then I think most importantly, and I've already mentioned this, writing always forces me to do a little bit more wide research in both the science and the non-science spaces. And so I think really importantly, for those thinking about writing, it's not good enough to just start writing and doing stuff. Of course, that's a great way to start and getting your ideas on paper. But if you want to capture an audience, I think the really important thing is writing about things that people care about in the spaces that they're expecting to read those things that they care about. So, when I'm writing for Signals, I'm thinking about the sort of both the stem cells and regenerative medicine academic field, but also a lot of the industrial players in it. And that's the sort of lens that CCRM puts it on. So really end up writing about, collaborations between industry and academia and the sort of cell and gene therapy space. With Hemisphere, that's the sort of experimental hematologist, but in particular, clinical hematology, and that's a space where a lot of the new tools that are coming out really need breaking down so that clinician scientists can really interact with the new tools and be able to extract as much information as rapidly as possible. So really trying to provide that sort of openness and discussion about new emerging things and science. And then my passion project, I guess, is the one around really trying to shape the way we handle education and training of scientists. Everything from parental leave to how we structure scientific careers where we're very risk averse because the sort of margins of success are so light. And then, this one was written originally in 2009, this bottom one. And this really just talks about that sort of change in the way science is structured, right? So we now have more houses, more babies, and more sort of partners when we're making these big life decisions about where to do science and how to do science. And I think that really shapes the way that we sort of structure scientists as a profession. And talk about that for years if you like. But I thought I'd just give a flavor of the things I write about. One thing that's important from a career perspective, especially if you're in the sciences, is that there are lots more sort of ways of trying to quantify that sort of contribution beyond the scientific papers and the citations themselves. And so you can pick any paper now, get these altmetric scores. It sort of summarizes where the story has been picked up and things like this, and who's writing about it. And that's a really effective way of showing the broader impact of your science, but also of other contributions as well. And then I thought I'd conclude by sort of pitching a few sort of challenges for scientists in particular writing about science. And really this sort of broke down into three quick areas. It's really challenging to get scientist readers outside of your own sort of space. So how do I get a bunch of physicists and chemists to read the sorts of things I'm writing about? That's really quite a challenge. Getting comments and really trying to understand how we push for change in the scientific record, especially if you're sort of peripheral to a particular domain. And then there's always the fear of repercussion, right? If you say something bad about some piece of work, somebody else might get angry about that and come after you. And that is a real thing. But I do firmly believe that we need to be open to open dialogue, as long as it's sort of polite and good interest. And so, for the sort of last slide of potential advice, I think it's really critical to think about what inspires or potentially enrages you, and maybe not make it quite about just yourself. And that's a really important thing for capturing that broader audience, understanding what the perspectives are from many different sort of stakeholders, and trying to then put pen to paper and put a logical argument together. And really, I think while you're getting that critical feedback is really the important thing in a lot of this sort of writing for the public and writing for scientists, is really to try and propose change. And I think that's really the only way. If you put something on paper that says it would be better if, and really trying to challenge people to think in that direction. And that's been a real feature of our writing over the years, on both the black hole as well as in hemisphere and signals. And with that, I'll stop, because I know we're on a bit of a tight schedule today, and I probably overran, but I didn't really track it. So hopefully that's okay.
Thank you so much, David. That was really great. Some really key considerations and great advice for the audience. So up next is Danielle Spice, who will highlight her strategies for simplifying complex scientific ideas into easily engaging concepts without losing accuracy. Dr. Spice earned her PhD from Western University, where she investigated early neural specification. She completed a postdoc at Sick Kids Hospital focused on kidney disease before transitioning into industry. Currently, Dr. Spice is a lead scientist at Apiary Therapeutics, an early-stage biotech company using genetically engineered stem cells and cell differentiation approaches to develop novel cell therapies. During her spare time, Dr. Spice engages with and educates lay audiences in biology concepts through her Instagram page at Science with Spice. Take it away, Danielle.
Thank you so much. So I think I'm going to have maybe a bit of a different kind of flavor to what David was presenting. And hopefully it'll be kind of a good back and forth about what you can do, where it's, David's talking more about like what you can do as a scientist trying to talk to other non-expert scientists. But not only do I have a background now of having a moderately successful and growing Instagram following as well as on TikTok, but also when I was at Western during my PhD, I was also working for the Center for Teaching and Learning there, where I was explaining to other TAs and educators on how to explain difficult concepts in science. So my background is mostly in talking about how to write scripts for educational content development, whether that's for an online space of Instagram or social media, but also in person and lesson design as well. So when thinking about our non-expert audience, we really have to first define who are we actually talking about. So we know that our experts are, our niche experts, but we may also have our general field experts. If you are an NK cell expert, there might be someone in your field who knows a lot about NK cells, but maybe they're more of a hematopoietic stem cell expert. But maybe we're talking to just scientists in general. But I think most of us, likely, since we're here, are probably talking about people who have an undergraduate level of science. So they're undergraduate science learners, either currently or they have a science degree, but mostly the general public. But then within the general public, are we talking about adults that have any sort of education background, or are we talking about K-12 science learners? And within that, that's really going to inform how we're going to actually go about our writing, is who we're trying to talk to. So one of the most important things in our writing, especially when we as experts try to engage with these lay audiences at any level, is defining our important terms. We forget, because we are so far down the rabbit hole of our own field, that each niche field has its own language. So as an example, if we're writing for field experts, we probably would say something along the lines of the transcription factor GATA6 is essential in promoting the differentiation of extraembryonic endoderm. But that doesn't mean a whole lot to general public adults. So we have to define those terms, specifically the ones that I've highlighted here of a transcription factor. This is a very niche biological term. So we need to then tell our audience whose function is to bind the DNA and promote the expression of a specific subset of genes. Similarly, for extraembryonic endoderm, that means nothing to a general population. So we have to define that term, a cell type that acts to support the embryo in the early stages of pregnancy. So we're still being accurate to what we're actually talking about. We're just giving a little bit of extra information. In order to kind of support our storytelling, there are some ways that we can do that. The first of these is to use examples. And I like to use examples to give context or to tell a story. So in this first example of an example, we have a single base pair change in the DNA of a protein coding gene can have huge implications. So here, what we're talking about, or what maybe I'm writing about, is the importance of point mutations and how that can have, or a detrimental effect, or maybe we're talking about, genetic disorders in general. And so as an example, I'm going to tell a story now of children who suffer from this debilitating and awful lethal disease have a single base pair change in the DNA that doesn't allow them to remove toxic byproducts. This is an example that gives context to what we're actually talking about. Just talking about point mutations between experts makes perfect sense, but to lay audiences, they don't know what a point mutation is. Similarly, we can give more context as a second example of, chemotherapy drugs target cells that are rapidly growing and dividing, but cause many normal non-cancerous cells, like the cells that grow your hair. So we don't have to, have this long-winded story or, this very large sentence like we did in the first example. We just need to provide that very basic amount of context. People know that your hair grows rapidly, and we're just telling them that these cells are also going to be affected by chemotherapy drugs. Lots of people know people within their lives that have lost their hair when they're on chemotherapy drugs, and it really just brings your audience with you on that story. My probably favorite way of bringing your audience with you is to use analogies. And using analogies can sometimes be challenging, and you really have to reflect on what is the analogy that you're going to use in order to not lose too much accuracy. You can use an analogy and then subsequently give your audience more information in order to potentially correct any sort of the shortfalls that you might have where your analogy doesn't perfectly fit what you're trying to get across. So as an example, we have GATA6 acts like a conductor, where we're directing genes to turn on and off, telling the cell how to become extraembryonic endoderm. Another example of an analogy, alleles are like using the words there, or there in a sentence. They all sound the same, but give a different meaning. And then subsequently in the next paragraphs or the next sentences, we can then give more information and then refer back to our analogy, again, to bring your audience along with you on the story that you're trying to get them to understand. I would argue, beyond giving context, one of the most important things when we're writing for lay audiences is, why should they care about it? We want to have some sort of hook, especially when we're thinking about in the social media space. We need to make sure that your audience is actually going to engage with your content. So we need to give that kind of upfront. So we want to understand stem cell reprogramming is essential because using induced pluripotent stem cells as a cell source reduces potential ethical, political, or moral hurdles. This is the why we care about stem cell reprogramming. And again, of course, we can go into more and more information afterwards. So this is finally, when you're kind of like building out what your article is going to look like, or what your video is going to look like, or your lesson, this, is directly from pedagogical or teaching theory techniques, where we want to scaffold the content that we're giving to our audiences. The important thing that you need to think about here is, once you've defined a term, you don't need to define it every single time that it comes up again. We can trust that our audiences know how to read and that they can, come along with that story with us. So, using the GATA6 example, we defined what a transcription factor is. We've defined what extraembryonic endoderm is early on in the article. Then we're going to use those analogies and examples to describe and define. So we talk about how GATA6 acts like a conductor. We're going to make sure that we give context and tell the audience, why do we care about this article about GATA6? Well, it's essential in understanding fertility and early pregnancy loss. This is an important thing for our audience. Finally, like I mentioned, we don't need to reuse defined terms. So we can actually use them in more field-specific language once we've already defined them. So as an example, in this new research article, GATA6 binding regions were identified and their associated genes were evaluated for an increase or decrease in gene expression throughout extraembryonic endoderm differentiation. That is perfectly fine once we've scaffolded our content to make sure that our audience knows what those words are before they see them in a more technical language. So hopefully I was right on time with what I wanted to tell you guys, but my main takeaway from this is that you are the expert in your field. And now it's just in becoming an expert in explaining your field to non-experts. Thanks.
Excellent. Thank you so much, Danielle. Our final panelist is Betty Zhu. She will address how to tailor content to broader audiences such as policymakers and the public. Dr. Zhu received her PhD in molecular genetics from the University of Toronto. She has over 10 years of experience working in research and science communication at Sunnybrook Research Institute, the Canadian Cancer Society, and the University of Toronto. Betty specializes in translating complex research topics into clear, compelling, and engaging content for diverse audiences and channels, and providing strategic communications support for leaders. Over to you, Betty.
Thank you so much, Joanna, and thanks for the invitation to be here today. I'm just going to get my slide up. Okay. All right, so I want to talk to you about how to tailor your content for broad audiences, and some of this will overlap a little bit with what Danielle was speaking about. But I think when you're trying to answer this question, the first thing, the most important thing that you have to do is to define who your audience is. So Danielle talked about, the different kind of audiences, adults versus kids, scientists versus non-scientists. But so I'm specifically going to focus on lay audiences, so people outside of science and academia. And even within that group, you can think of a lot of different stratifications, right? Like how old someone is, what lived experiences they might have, their different needs and preferences. Is it an internal audience? So are you communicating this to people within your own department or faculty, or is it an external audience where you're trying to reach members of the general public? And for me personally, in my experience, some of the different audiences that I've specifically written materials for are people like donors, alumni, policymakers. So going back to David's point earlier about advocating, so reaching those people in leadership positions at universities, at hospitals, at the government funding level, corporate partners, students, patients is a really important population that we often communicate with, and they will have, unique lived experiences and different needs and preferences. So it's really important to know who your audience is, because knowing who they are will define the rest of your communications plan. So I'm not sure how many of you know what a communications plan is, so I just wanted to back up a little bit. And I asked ChatGPT to tell me what a communications plan is. And the definition it gave me is actually pretty good. So it's a strategic document that outlines how information will be shared with specific audiences to support a project, initiative, campaign, or organization's goals. So the idea of this plan is to really ensure that everyone is working with the same messaging and that this messaging is delivered in a consistent, timely way, and it's aligned with the outcomes that we're all working towards. So some of the key components of a communications plan are the goals and objectives, your target audience, key messages, channels, tactics, timeline, and evaluation. So all of these things can really be slightly altered and defined based on who that target audience is. So for example, what are your goals and objectives, right? So if you have one really exciting new research finding you want to share, you might have different goals and objectives to share that depending on who you're trying to reach. So maybe for the general public, you want to inform them, educate them about this new research finding. But maybe for kids, you want to make it a little bit more fun and entertaining. And so you might use a different strategy or tactic to achieve that objective. Whereas you might, leverage this new finding in a communication, in a memo to policymakers to try and persuade them to increase funding for CIHR or something like that. So depending on what the goals and objectives of your communications are, you're going to have different tactics to achieve those goals. So if you want to inform, educate, explain, you might be working on a news story, a Q&A, a backgrounder, versus entertain, you might be working on a TikTok video or an Instagram reel or something that's a little bit more lively. Persuasion, so these are often, for example, op-eds or opinion pieces or what we call thought leadership pieces that leaders might write in like the Globe and Mail or, the Hill Times or those really big newspapers. And if you want to tell a story, maybe about the people behind the science, you might be looking at a profile. So that's not to say that, you can't have multiple objectives. And of course, your piece can maybe both persuade and inform. But this just gives you an idea of how, really having a clear goal at the outset will really help you narrow down what is the best channel of communication and the best method of communication for your audience. And then we need to think about what are our key messages, right? So you have this really new, exciting research you want to share. What are the 3 key takeaways you want your audience to walk away with from this finding? And that message might change a little bit, right? So if it's a really exciting new finding, scientists have discovered like a new use for these stem cells, right? That might be the overall key message. But if you're communicating to donors, for example, you might alter that key message a little bit with something like, thanks to your generous support, scientists at the Stem Cell Network have blah, 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 right? Or if you're talking to policymakers, you might say, thanks to the government's investment in the Stem Cell Network, we've been able to achieve X, Y, and Z. So your overall key messages should be the same, but you can kind of tweak them a little bit to adapt them and make them relevant for your specific audiences. And when you're thinking about key messages, it's generally best practice to keep it to 3 key messages and no more than that. And lastly, who your audience is really impacts what channels you use and what tactics you use. So one of the really important things you have to keep in mind is you need to meet your audience where they are. So, if your audience is a younger demographic, maybe social media is the best way to use them. If your audience is industry partners, corporate partners, maybe LinkedIn is the best way to meet them. So thinking about, which of these channels is your target audience most likely to read, to listen to access, will determine what you should be communicating and how you should be communicating it. And then tactics are how you reach your audience. So, for example, if you just want to target the general public really broadly, maybe your tactic is to send out pitches to journalists at the CBC, CTV, Globe and Mail and things like that. But if you really want to target, incoming graduate students to recruit them to a stem cell network event, maybe you're more on social media and you're creating content for Instagram and TikTok to reach that younger demographic. So really thinking about, holistically as your content comes together, how are you adapting the same key message to reach different audiences on different channels and using different tactics will really help ensure that your message is reaching the right people in the right way. So I just want to use the last bit of time to walk you through a bit of an example. So I recently wrote this story for the Temerity Faculty of Medicine's website, and it was about a research collaboration that had just received a green light from Health Canada to conduct the first in human clinical trial. So this was a new type of nanoparticles called porphosomes, and it's just a really exciting finding. So I wrote a story for the Temerity Faculty of Medicine's website, and the key messages for this that I wanted readers to take away from the story was one, that a team of Toronto researchers has gotten Health Canada approval to test a homegrown innovation in a world first clinical trial. So the kind of key words we want people to take away from that, it's a team of Toronto researchers. They have Health Canada approval. It's a homegrown innovation. So we really want to emphasize that this is a made in Toronto innovation and this is a world first clinical trial, right? So just that first sentence alone gives you why it's relevant, why it's impactful, and why you should care. The second key message talks a little bit about what the innovation is and what potential it has to improve cancer management. And then the third key message is something that as an institution, we really wanted to highlight was that this was enabled by collaborations between University Health Network and the University of Toronto's Temerity Faculty of Medicine and Leslie Dan Faculty of Pharmacy. So really highlighting how we're working, across institutions, across faculties to get these really impactful innovations from the lab to patients. So this was the key messages, and this was the website with the story was what we were really hoping to drive different audiences to through different channels and tactics. So to try and reach current and prospective students, one of my colleagues in the pharmacy faculty made a really awesome Instagram reel, and he shared that on Instagram and tagged all of us. So it came up on all of our feeds. And we know that our Instagram channel is where a lot of new students, prospective students come. So we wanted to have a really engaging short video clip to showcase to them, this is the type of research we're doing. This is the type of research that you can be involved in. And then for our faculty, alumni, and industry partners, we had a post on LinkedIn that really highlighted kind of the innovation aspect of it, that highlighted the cross collaboration. And that went on in LinkedIn where we got tons of really great engagement as well. So if we were to adapt this for something like donors, you might want to include this in an impact report to a donor who maybe has given money to support cancer research. And you might tweak that to say, thanks to your generous support, researchers at University Health Network and U of T have, just gotten Health Canada approval. Or you might adapt it with, in a different memo to hospital and university leadership that shares this really unique finding, but really moving that key message about collaboration up to the top. So hopefully gives you a bit of a sense of how you can take, one really exciting science idea or concept and adapt it to multiple different platforms to reach and engage different audiences. And I just want to end by just reminding you that, effective communication really means delivering the right message to the right audience. So if you're writing something, if you have something that's really cool, but it's not reaching the right audience, then that communication isn't really working. So really thinking about not just the content you're creating, but also how you're getting it to the people that you want to see it is really important as well. And something to keep in mind, at the very beginning when you start thinking about what you're going to write, what you're going to create. Thanks for your time, and I will stop sharing.
Thank you, Betty, for closing out our talk.